Hello, class. We are going to dive into Chapter 11, Blood and Blood Vessels. It's a doozy of a chapter. There's a lot to it. And this week you have two chapters you're responsible for, all dealing with the heart, blood, and blood vessels. So pace yourself and hopefully find this lecture helpful in learning that new material. Let's go here. Pull the PowerPoint up and let's get started. Here are the learning outcomes that are already listed for you in your weekly folder. Let's just define some of the terms that are related to the cardiovascular system. First, you have the fluid compartment or the blood. Then you have all of your tubes or your vessels. We call them the blood vessels. These are capillaries, arteries, and veins. And then you have a pump that's pumping all of the fluid through the vessels. That's your heart, obviously. All right, so your blood, the main function is to distribute oxygen, carbon dioxide, blood cells, all around the body. It's also delivering nutrients and hormones. It's transporting waste away from the periphery and getting them to wherever it needs to go, either to the kidneys or to the lungs. It's also assisting in temperature regulation and defense against disease. Your blood vessels are distributing that blood throughout the body, and the heart propels the blood and maintains blood pressure. So let's talk about the overall functions of blood. Transportation, it transports nutrients, carbon dioxide, oxygen, hormones, wastes. It regulates the pH. The blood has buffers in it to help regulate the overall blood pH, maintaining a 7.35 to 7.45 blood pH. It restricts fluid loss at an injury site, so it forms a clot to help stop hemorrhaging. It's also going to defend against toxins and pathogens. It's a main mechanism for our immune system to mobilize throughout the body, and it also helps to stabilize our temperature by absorbing heat and distributing blood flow to different areas of the body. Let's talk about our blood composition. It's called a fluid connective tissue. We learned that way back in Chapter 3. It consists of the plasma, which is the fluid or liquid portion, and then the formed elements, which are the cells. On average, it's about 5 liters of blood in your body, a little bit more for males, a little bit less for females, and whole blood is the term for the blood that's removed from the body, but plasma can then be fractionated or separated for its platelets or plasma, etc. Roughly the same as body temperature. It's 5 times thicker than water, and again, it has a slightly alkaline pH. 7.35 to 7.45 is the range. Here you see all the formed elements. And plasma composition. We're going to go through these individually. Our plasma is the majority of blood, a little bit more than half, 55%, transports all of the organic and inorganic molecules, the formed elements, as well as heat energy. It's composed of mostly water, has some plasma proteins and other solutes inside of it. Those plasma proteins are things like albumins, globulins, which include our antibodies and transport proteins. Also fibrinogen, which is used for clotting. It forms strands of fibrin, which is that kind of tough, thick, strandy stuff that makes up our blood clots. The other solutes involved in our plasma are our electrolytes: sodium, potassium, magnesium, calcium, chloride, phosphate, sulfate, and bicarbonate. We also have organic nutrients like lipids, carbohydrates, and amino acids, and organic waste like urea, uric acid, bilirubin, ammonium ions, and creatinine. There you see the basic breakdown of our plasma. Now we get to the formed elements. Our formed elements are a little bit less than half. A hematocrit, or packed cell volume, PCV, is basically the percentage of whole blood volume that is just formed elements. The average is, again, little less than half. Androgens in males are going to stimulate red blood cell production, which gives them slightly more. So our hematocrit, or our formed elements, is mostly 99.9% red blood cells, small percentage is white blood cells, and a very small percentage is platelets. Our red blood cells, or erythrocytes, are the most abundant blood cell. Their function is to carry oxygen. White blood cells are part of our defense mechanism. They're also called leukocytes. And platelets are just little cell fragments. They're important in the clotting process. There you see them. Their percentages, roughly what they look like. We're going to get into all of that right now. You could pause the video here, go back and answer these 3 questions. Let's talk more specifically about red blood cells. Red blood cell count is the number of red blood cells per microliter or cubic milliliter. Roughly, again, more in males than in females. That's going to be pretty much the trend throughout all of A&P. It's always a little bit bigger, a little bit more in males. They tend to be bigger. They tend to need more blood. red blood cells make up about 1/3 of all of the cells in the human body. So if you took all your body, piled all the cells and divided them into categories, about 1/3 of all the cells, just blood. They're biconcave discs, which means they're kind of imploded on either side, make a very thin center, thicker outer margin. As you can see here, that cross section, thin in the middle, thicker on the sides. Biconcave disc. When you look at them under the microscope, they kind of look like this, just little purple dots. 450 is about the magnification that you would get in lab, unless you were using oil immersion. So this is pretty much what you would see. You can see a little bit of those little fragments in there, which are platelets. They don't have any white blood cells on that slide, but we'll see them in a little bit. So why are the blood cells shaped like they do? It's all about the surface area to volume ratio. The oxygen gets bound to the hemoglobin within the red blood cells, and that greater surface area allows for a faster exchange of oxygen. So it's just about efficiency. The red blood cells can also stack on each other because of their shape. That's going to allow them to flow nice and easily through those narrow passageways and capillaries. They also have flexibility because of their shape and because of the fact that they don't have a nucleus, they're anucleate. They can kind of squeeze and squish through tight capillaries. Here you can see little stacks of them squeezing through blood vessels. So let's talk about some features of our red blood cells. Again, they're designed to carry oxygen. They have very few organelles, including no nuclei. They're anucleate. They cannot divide, and they cannot repair themselves because they don't have all of those cellular machinery to do that. Their lifespan is less than 120 days. So less than 120 days, your blood cells they're constantly being recycled. They're filled with hemoglobin, which is a protein, and the hemoglobin, again, is more in males than it is in females. There you see it. Here is a hemoglobin protein. The hemoglobin protein has 4 hemes on it. Heme is kind of like a binding site for iron, and that's what you see, this little red dot on the heme is iron. So hemoglobin is composed of 4 globin subunits. Each subunit has a heme, and that heme holds an iron, just like I was just showing you. The blood with the oxygen bound to hemoglobin is called oxyhemoglobin. It's very bright red, and blood with lots of hemoglobin that's not bound to oxygen is called deoxyhemoglobin, and it's not very bright. It's more of a darker red. So here is the oxyhemoglobin. Red blood cells are continually produced and recycled because they only have a lifespan of 120 days. Your body has to constantly renew and create them. About 1% of your blood supply is replaced every day, which means 3 million new blood cells enter the bloodstream each second. Wowza. With that short lifespan, the plasma membrane basically ruptures and the cells engulfed by macrophages, which are the little Pac-Men that float around your blood, gobbling up debris. They're broken down and recycled by the liver and the spleen or the red bone marrow. You can pause the video here, go back and answer these 3 questions. Let's talk about blood type. How many of you know your blood type? Probably not many of you. Usually in my typical class of about 50 students, I have about 1/3 of my students or less that know their actual blood type. A couple ways you can learn your blood type. You can join the military. They do blood typing there. You can donate blood. They will tell you your blood type, or you can look at your parents and figure out their blood types and then deduce your own blood type from theirs because it's inherited. Your blood type is determined by antigens. Antigens are little surface proteins on the outside of blood cells. They're specific to blood cells, and they will induce an immune response. So your plasma membrane of all of your body cells has antigens on it. Your immune system can recognize those cells as yourself. So it's like, hi, I'm a Jamie cell, I belong here. Your blood type is determined by those antigens that are on your red blood cells specifically. So there are specific antigens that are only on red blood cells. They are A, B, or D antigens, or all 3, depending on what your blood type is. So the plasma, the liquid portion, contains antibodies. The antibodies respond to the antigens. They can attack foreign antigens. So if there's another organism or another blood type in my blood for some reason, my antibodies are going to recognize that those surface antigens don't say, hi, I'm a Jamie cell. They might say, hi, I'm a foreign invader cell that wants to kill you, whatever. Our antibodies are going to attack that, kill it, and protect the rest of the body. So here you can see what I mean when I'm talking about antigens. They're little proteins on the outside of the cell. Type A blood has type A antigens. Type B blood has type B antigens. Type AB blood has A and B antigens. And look at blood type O. There are no O antigens. O is just the absence of A and B. So look here, if you have blood type A, you're going to produce anti-B antibodies. If you have blood type B, you're producing anti-A antibodies. If you have blood type A, AB, well, guess what? You don't have any antibodies against blood types because you have both of those antigens. But if you're blood type O, you're going to produce anti-A and anti-B antibodies. So this is where cross reactions come into play. If you're exposed to another blood type other than the type that you have, the agglutination or clumping process will begin. What this is going to do is it's going to basically cause all of those antibodies to agglutinate or clump around the foreign blood, and that's going to cause those blood cells to rupture. This is called a cross reaction. You can get very, very sick. This would happen from maybe a transfusion of the wrong blood type. There you see your surface antigens and opposing antibodies, that agglutination occurring, breaking up those red blood cells in the process called hemolysis. Let's talk about complete blood. A complete blood type is basically not just the A, B, or AB, but we're also talking about the rhesus factor or Rh factor. It can be positive or negative. This is the D antigen that I was mentioning before. So an Rh positive person has this Rh surface antigen on their blood cells. An Rh negative person does not. So this is going to influence how your blood type is reported. You're either A positive or AB negative or O negative, right? That positive or negative is telling you whether or not you have that Rh factor. So this is just the overall distribution of blood types throughout. Most people are Rh positive. It's fairly rare to be Rh negative. And interestingly enough, your heritage can also influence what type of blood you have. There are more common blood types in different populations. So for instance, I'm blood type B, and I'm B positive, and obviously I'm Caucasian. So only 20% of Caucasian Americans have my blood type. It's pretty rare, which is why the Red Cross calls me all the time to tell me that they need my blood. But look at this. People that have blood type O are very common, almost 50%. Look at Native North Americans, 79% are all type O blood. So it's strongly related to heritage. If you look at AB blood, it's relatively a rare blood type. B is also a relatively rare blood type. Blood type A and blood type O are fairly common. So let's talk about how you do a blood typing test. You may get to do one of these in lab. You basically take a drop of blood and you mix it with an antibody solution and you look for clumping. You look for that agglutination to occur. That cross reaction is also called a transfusion reaction. No transfusion reaction would tell you that the blood types are compatible. If you see clumping, that means that they're incompatible and that you can't donate. So here you see this clumping. If you have A positive blood and you expose it to the anti-A antigens, it's going to clump, right? Because it's attacking that anti-A. If you have A blood and you expose it to anti-B, well, there's no clumping, which means it's compatible. If you have A positive blood and you expose it to that anti-Rh, it's going to clump, which means it's compatible. So we're looking for the compatibility. The positive test is going to indicate that they're not compatible. So anti-Rh is going to attack that Rh antigen. You cannot give whatever this anti-Rh serum to someone that has Rh positive. Same thing here. Someone that's B positive, they're not going to have a response to the anti-A antibody because they already have the anti-A antibodies. But if you give them anti-B, you're going to see clumping, so on and so forth. So anti-A and anti-B antibodies are both present at birth, and the anti-Rh antibodies are only formed if an Rh negative person is exposed to an Rh positive blood. So in other words, if you are Rh negative, you don't have to worry about a transfusion reaction with the Rh factor unless you've already been exposed. So you can pause the video here, go back and review. Let's talk about one instance that may be life-threatening. It's called hemolytic disease of the newborn. Basically, the surface antigens on the red blood cell, as I told you, are genetically determined. So a child inherits genes from both parents, and the genes that the child has may be different than the mother or the father. Really, that only pertains to the mother, though, because the mother is growing the baby. So during pregnancy, the mother's antibodies may cross into the placenta. If the fetus is Rh positive, and the mother's Rh negative, the mother can produce Rh antibodies and attack the fetus. The result is called hemolytic disease of the newborn. If an Rh negative mother has never been exposed to fetal blood until delivery, there's no problem. But with the second pregnancy, now that that mother has been exposed to the Rh, she can now make the Rh antibodies. Now she may attack the second fetus, the second pregnancy, because she's now producing those Rh positive antibodies. So that process is called sensitization. You have to be sensitized to the Rh factor by being exposed at least once. So in future pregnancies, those anti-Rh antibodies can cross over into the placenta and kill it, basically. The maternal anti-Rh antibodies can cross, destroy the fetal red blood cells. The dropping red blood cell count can ultimately cause the fetus to abort or miscarry. In science, we call it a spontaneous abortion. It's a miscarriage, but that's the technical term. That's why I use that word. So there's basically a shot that mothers can take if they're Rh negative. It's called RhoGAM. RhoGAM is going to basically desensitize the mother to those Rh antibodies in the case that her unborn baby may be Rh positive. So Rh negative mothers who are pregnant with their second child will have to or should be aware of the fact that they need to take RhoGAM shots in order to prevent erythroblastosis fetalis, which is basically the fetus dying because of the attacking mother's antibodies. So here's just a visual of everything that I was just explaining. That first pregnancy, she has Rh negative antibodies exposed to the Rh positive fetal blood, and the hemorrhaging and detachment of the placenta at birth causes that exposure or sensitization. So then during the second pregnancy, that mother already has those antibodies formed, and when it interacts with the fetal circulation, that baby could suffer from hemolytic disease of the newborn. So you can pause the video here, go back and review. Let's talk about the shared properties of white blood cells. White blood cells circulate in the bloodstream for a short period of time, then they go back to their loose or dense connective tissue. They kind of trickle out like little surveyors, and then they go back to where they live, and then they go out, and then they go back, right? They're activated in the bloodstream, so they can squeeze through special cells. They can attach to, and they're attracted by chemical stimuli. It's called chemotaxis. They're kind of like sharks sniffing out the blood in the water. It's kind of how white blood cells work. They sniff, they can tell when tissue's been damaged, they can tell when an invader's been there, they can tell when the immune response needs to be activated, and so they migrate to that area. Neutrophils, eosinophils, and monocytes are all phagocytes. Phagocytes are eaters, like little Pac-Men, going through and gobbling up debris. Here you can see what a white blood cell looks like under the microscope compared to the red blood cells. You have leukocytes or lymphocytes, you have your monocytes, and you have your eosinophils and your basophils and neutrophils all in there doing their own little jobs. So let's talk about what these things are. They're called granular leukocytes. Granular leukocytes are the most abundant. And the reason why they're called granular is because they look kind of spotty under the microscope. They look like they have grains inside of them. These are the neutrophils, eosinophils, and basophils. Neutrophils are the majority. So when you're looking under the blood slide and you see a white blood cell, it's more than likely a neutrophil, and you might even see a few of them in there. Eosinophils or eosinophils are not quite as common, same thing with basophils. So here you see them, and overall functions. Neutrophils are phagocytic. They go through and gobble up any debris, any waste products, any bacteria. Eosinophils are specifically for parasitic infections and allergies, and basophils are responsible for assisting in that inflammatory response to assist with histamine and heparin release. Agranuloleukocytes are very few and far between. You have monocytes, which is a very small percentage, and lymphocytes, which is a larger percentage. Lymphocytes, these are the T cells and B cells that you've probably heard of when you've heard of immune system. So here you see them. Monocytes are phagocytic, and lymphocytes are providing specific defense against specific pathogens. So let's talk about white blood cell counts. Pathogenic infections can cause changes in the numbers of white blood cells. In other words, if your body is undergoing some type of invasion, cancer to strep throat to a common cold, whatever, your white blood cell count is going to be elevated in order to respond to that. The differential count of white blood cells could indicate the type of infection that you have. So here are some questions you can go back and review for that little section we just covered. Let's talk about how these cells are made. They're all formed from the red bone marrow, originating from what's called a hemocytoblast, which is your stem cell for blood. It's a process called hematopoiesis. Hematopoiesis will, always begins with the hemocytoblast, and that hemocytoblast can either produce a lymphoid stem cell or a myeloid stem cell. So the lymphoid stem cells will develop into lymphocytes. They typically will go out into lymphoid tissues like the thymus or lymph nodes, and they will mature there. They will finish their growing there. Myeloid stem cells will develop into other formed elements, other cells. Something called a colony stimulating factor, or CSF, is going to determine how and what type of cells these myeloid stem cells differentiate into. So here you see the hemocytoblast. It can either be myeloid or lymphoid. Lymphoids develop eventually into a lymphocyte, and that development happens within the lymph tissue, like the thymus or like a lymph node. All of the other cells follow this similar pathway in the red bone marrow from a myeloid stem cell, ultimately either to a monocyte, a neutrophil, a eosinophil, basophil, platelet, or erythrocyte. So red blood cells are under hormonal control. Erythropoietin, or EPO, is released from the kidneys when oxygen levels are low or hypoxia is occurring. This is when things like anemia is going on, maybe declining blood flow to the kidneys, decreased oxygen from the lungs, maybe you're living at high altitudes, and finally damage to the respiratory surface of the lungs, all stimulating your bone marrow to make more red blood cells. It's called erythropoiesis, or making red blood cells. EPO is also going to stimulate stem cells to increase their manufacturing of red blood cells. Platelets are actually cell fragments. They break off from a huge cell called a megakaryocyte. They're basically little flattened discs that function in blood clotting. They're continually replaced every 9 to 12 days before they're gobbled up. Usually that recycling happens in the spleen. Our spleens are a major recycling center for blood. Oh, sorry. You can pause the video here, go back and answer these 3 questions. You probably love it when I say that, don't you? Let's talk about the clotting response. Clotting is called hemostasis. Not homeostasis, hemostasis. So hemostasis is the process of stopping blood from flowing out of a vessel, out of a damaged vessel. So there's a cascade effect that occurs in the clotting process, or in this hemostasis process. There's 3 phases involved in this cascade. There's a vascular phase, a platelet phase, and a coagulation phase. Let's talk about the vascular phase. This is about 30 minutes after the injury. There's 2 major events that occur. First, you have your endothelial cells, or your kind of lining cells, that contract and reveal the basement membrane. And then the smooth muscle is going to contract also in something called a vascular spasm. So in other words, everything's opening up after the injury happens. And the reason that all of that, I'll flip back here, the reason all of it opens up and retracts is so that the blood flow can wash out that area, get any pathogens, any invaders out, and start attaching platelets to the area for a clot to form. So as soon as those platelets start to attach, now we're in the platelet phase of hemostasis. The sticky endothelial cells of that basement membrane start to collect platelets inside and outside. And then there's clotting factors that are released by the platelets. This is positive feedback that's going on here. So once the platelets start sticking, they're kind of cheering each other on and saying, okay, let's keep clotting, let's keep clotting, come on guys, come over here, let's keep clotting. And this is all going to basically stimulate that vessel to contract back together. The coagulation phase is usually 30 seconds or more after vessel damage. So this is later, again, but we're still talking seconds. This isn't like a day later, right? And this is when that clotting complex requires something called a clotting factor that's floating around the plasma. There's several proteins, calcium is involved here, vitamin K is involved here. They're all clotting factors to encourage basically the fibrinogen to be converted into fibrin. That's the big kind of purpose here. Fibrinogen is a clotting factor that's floating around your blood. It's made in the liver, and as those platelets are forming there and they're kind of forming this little clot, fibrinogen is eventually converted into fibrin, and that's kind of the final seal of the clot. And that fibrin network is basically trapping the blood cells and the platelets in, and that damaged area is now sealed. There's two pathways to how this happens. It happens on the outside, which is called the extrinsic factor, and it happens on the inside, which is called the intrinsic pathway. So the extrinsic pathway is controlled by tissue factors. You know, if your damaged blood vessels here, and it has this big kind of hole in it, the extrinsic pathway is going to start patching from the top. The intrinsic pathway is going to patch from the bottom, and then they kind of meet in the middle for this common pathway. So the common pathway is now, once the two extrinsic and intrinsic pathways come together, and there's a factor called factor X, which is activated once this common pathway is ready to go. Factor X forms something called prothrombinase. Prothrombinase is an enzyme. It converts prothrombin into thrombin. And then thrombin is the molecule that converts fibrinogen into fibrin. And fibrin is that fibrous clot that finally seals the wound. After a few hours or so, the clot will begin to contract. This is basically pulling our tissues back together. This is why scabs eventually feel tight, because the clot has retracted and pulled that tissue back together. And this can be kind of itchy feeling also. Here you see the pathway. Vascular phase, everything's contracting, vascular spasm, extrinsic pathway, intrinsic pathway, and then in the middle you have our common pathway activated by factor X, converting to prothrombinase. Prothrombin to thrombin is converted, and thrombin converts fibrinogen to fibrin, and fibrin is finally the end step of clot formation that will allow the clot to retract and pull those endothelial surfaces together. Blood clotting can form inappropriately. They can happen in the bloodstream where there's no injury. That would be called an embolus, where you have this drifting blood clot. It can damage small blood vessels. It can block blood flow to an area, causing tissue death. A sudden blockage is called an embolism. Or tissue damage can actually result in something called an infarction or an infarct. This can happen in the brain, and it's called a stroke. Or it can happen in the heart, and it's called a myocardial infarction or an MI or a heart attack, right? Just blood clots floating around. Imagine this big embolus floating around, blocking the blood vessel. Why does it form? Well, it can form because there's too many clotting factors in your blood. It can form because your body metabolism is thrown off by an unhealthy diet, hereditary factors, etc. There's lots of reasons why these would form. A thrombus is a blood clot that's attached to a vessel wall where there's no damage. So this can form where platelets are attracted to plaques. Plaques are related to large quantities of lipids in your blood, so they kind of attach to the walls of your artery and your blood vessels, and then these thrombi can attach to those plaques, and they can dislodge and become an embolus and float up and cause an infarct. Or they can just block an area like this, right? So the clots, once they're formed, will eventually dissolve, and we all know this just from ever damaging our skin in life. It's a process called fibrinolysis. Begins with plasminogen being converted into plasmin, and that plasmin is basically eroding the clot. You can pause the video here, go back and review. So let's talk about some ways that we can diagnose blood disorders. First, you have to get the blood. How do you get the blood? Primary method is through venipuncture. Typically, the blood's collected from a superficial vein. Superficial veins are very easy to get. The blood pressure is relatively low, so it's not going to squirt all over the place and damage. And the walls of veins are typically thinner than the walls of arteries, so it's easier to obtain. Do you like needles? So, some nutritional blood disorders. Iron deficiency anemia can be caused by insufficient iron. It is more common in women because our iron reserves are lower than in men, and overall, our red blood cells can't transport as much oxygen. There's also pernicious anemia. I can never say that one. And that's because of a deficiency in vitamin B12, preventing normal stem cell division in the bone marrow. And so your red blood cells are going to be very big and oddly shaped. So they're not going to carry oxygen as efficiently. Finally, there's clotting disorders. Typically, clotting disorders can be related to insufficient calcium or insufficient vitamin K. Both calcium and vitamin K play important roles in the clotting process. They act as those kind of clotting factors. You also have hemophilia. This is a congenital blood disorder. So this is not something that can be caused by improper nutrition. This is an inherited blood disorder. It affects about 1 in every 10,000 people, mostly males. It's an X-linked trait, sex-linked trait. So it's carried on the X chromosome, affecting mostly males, caused by missing or reduced production of specific clotting factors. In severe cases, bleeding with very minor contact around joints and muscles can occur. So individuals with hemophilia have to either take oral clotting factors to boost their clotting factors in their blood, or they have to live within proximity of a hospital so they can get help if they do suffer an injury. Thalassemia is a characteristic group of inherited blood disorders, typically where there's an issue with the hemoglobin. Hemoglobin's either improperly formed or not functioning properly. And finally, sickle cell anemia. Sickle cell anemia results from a mutation that's affecting one amino acid of the whole polypeptide chain. The red blood cells then become sickle-shaped, and they release, fixing my chair here. They release oxygen, and therefore they don't have that typical disc shape. They're more shaped like a crescent moon. They're very flexible, easily broken. They can actually get stuck in blood vessels and block blood flow. So that can be very dangerous. Now, some people have 2 copies of the sickling gene. If you have 2 copies of that sickling gene, then you're going to have sickle cell disease. If you have 1 copy of the sickle cell gene, if you're what we call heterozygous, you have what's called the sickling trait. Sickling trait is kind of a good thing. It means the majority of your blood cells are just normal shaped. You might have a few that are sickle shaped, but it provides resistance. Kick the wall here. It provides resistance to malaria. So people that live in the malaria zones, like in Africa and India, can have resistance to malaria by having just 1 copy of the sickle trait. Kind of a cool thing there. There's those sickle cell red blood cells. So let's talk about some blood infections. You have bacteremia. This is just when bacteria is circulating in your blood. It doesn't multiply there, but it's circulating. Viremia is viruses circulating in the blood. Viruses don't multiply unless they're inside of a cell, so it's not multiplying there either. Sepsis is a widespread pathogenic infection. Septicemia is blood poisoning. This is a sepsis specifically in the blood. The pathogens can multiply in the blood, spread throughout the body, very dangerous. Septicemia. Malaria is a parasitic condition caused by a paramecium, or a protozoan rather, called Plasmodium, and it kills millions of people every year. It's transmitted by a mosquito, something as simple as a mosquito. It's more common in tropical countries, like I've said, and as I mentioned, individuals who have that sickle cell trait or sickling trait, just 1 copy of that sickle cell gene, are immune to malaria, kind of a benefit. Basically, what happens in malaria is the parasite invades liver cells, and so the liver cells get big and they start to break off, and those little fragments of liver cells affect red blood cells. And overall, you get cycles of fever and chills, and reinfection can occur. So the dead red blood cells can block vessels, and it can be very dangerous. There's our mosquito. There's also cancers related to our blood. Leukemias are cancers throughout our blood dealing with our white blood cells. They can affect our blood-forming tissues and spread throughout the body. Myeloid leukemia is when the abnormal granulocytes are forming in the red bone marrow. Lymphoid leukemia is involving the lymphocytes in their stem cells. Symptoms will generally appear when there's abnormal white blood cells in circulation, and the symptoms can be very general. You can just get sick a lot. Oh yeah, I get sick a lot, but that's because I work around kids, or that's because I don't wash my hands, or whatever. So if you're noticing that you're getting sick more than you should be, I'm not saying you have cancer, but keep in mind that that's one of the general signs of leukemia, is just an inability to resist infection because your white blood cells are damaged. I just don't like the way this camera's sitting. Sorry. Oh, well. I'm like way down here now. Oh, well. All right. Here are your abnormal white blood cells. You can see they don't look right, and there's an abnormal ratio to them. So there are degenerative blood disorders as well, where you have bacterial toxins that can activate steps in that clotting process, causing what's called disseminated intravascular coagulation, or DIC. The fibrinogen is converted into fibrin within circulating blood. That can be very dangerous, right? Small clots can float around, block blood vessels, and the result is ultimately uncontrolled bleeding because your liver is increasing its fibrinogen concentration or fibrinogen secretion, and there's nowhere for it to go. So it's very dangerous. You can pause the video here, go back, and answer these 3 questions. Let's talk about blood vessels and their circuits. Are you knocking on the door?
Yes.
What do you need?
You want lunch?
Yes, please. Can you just heat me up that soup that we had?
Oh.
Or that. Thank you. Thank you.
So I thought
What a good husband.
Oh, you're still recording?
I am.
Oh, great.
At least he was wearing pants. So let's talk about blood vessels and their circuits. So we have 2 main circuits in our body. We have the pulmonary circuit. Food smells good. Pulmonary circuit, which is carrying blood from the heart into the lungs. And then we have the systemic circuit, which is carrying the blood to the rest of the body. So there's 2 major pathways for this to occur. You also have blood being carried away from the heart in the form of arteries or efferent vessels, just like we had in the brain, right? And then we have blood returning to the heart in veins or afferent vessels. And then we have capillaries, which are connecting the smaller arteries and smaller veins together. They have very thin walls to allow for exchange of nutrients. Here you see these circuits. I strongly suggest doing this. This is how I would study it. Take note cards or Post-its. Write each individual word, like right atrium, right ventricle, pulmonary artery, capillaries, and the lungs, etc., on Post-its or note cards, and see if you can put them in order. That's a really great way to study this, or just by tracing it and saying it out loud. Right atrium, right ventricle, pulmonary arteries, capillaries of the lungs, pulmonary veins, left atrium, left ventricle, aorta, right, etc., etc. Going through like that. So when the right side of the heart is basically getting the blood back from the rest of the body and taking it out to the lungs, and the left side of the heart is getting that oxygenated blood back from the lungs and pumping it out to the body. And then it goes back to the right side, then it goes to the lungs, then it goes to the left side, then it goes to the body, and so on and so forth. So let's talk about how these arteries and veins are formed and their overall structure. You have the tunica intima. This is the innermost layer. Then you have the tunica media, which is the middle layer, and the tunica externa, which is the outermost layer. The tunica intima has an endothelial lining. It also has an internal elastic membrane, so it can help it recoil and expand and contract just a little bit. Your tunica media, I don't like this. Oh well. Tunica media is the middle layer. It has concentric rings of smooth muscle. This is the muscular layer, media muscle. Muscular layer of our arteries and veins. It allows for constriction and dilation. The arteries actually have an extra external elastic membrane for more recoil. Then you have your tunica externa. This is the outermost layer. This is the connective tissue sheath. This is the kind of outer covering. Veins tend to have a thicker tunica media, and the connective tissue fibers help bind it to adjacent tissues so they can kind of sit and be held in place. The arterial tunica externa has mostly collagen fibers, making it very strong, resilient. Here you can see an artery, more muscular layer, tunica media. A vein has a much thinner tunica externa, and check out the lumen of these things. The vein is much more relaxed. The artery is very spherical shaped because the blood pressure in the artery is much higher. Here you see just a chart from your book comparing arteries and veins. Let's talk about different types of veins. You have large veins, which have all 3 layers, intima, media, externa. These are typically the superior and inferior vena cava and some of the major branches off of that. Medium-sized veins have a relatively thin tunica media, very small smooth muscle cells, and a thicker tunica externa. And then you have venules. Venules are the smallest venous vessels. So you see the three. And then arteries can be elastic or muscular, or they can be arterioles, which are the small ones. Large elastic arteries, this is like the pulmonary trunk, the aorta, and the major branches off of those vessels. These are very elastic. They're very capable of stretching and recoiling. The muscular arteries, these are the ones that are going to dilate and constrict in response to sympathetic and parasympathetic input. These are medium-sized arteries. And then the arterioles is where the tunica media only has a little bit of muscle. So these are not going to dilate and constrict as much as those muscular arteries do. And notice the arterioles don't have a tunica externa. So let's talk about blood flow pattern. Arteries carry blood away from the heart. They branch into smaller arteries, and they branch into arterioles, and then they lead the blood to capillaries. From the capillaries, the blood's going to flow through little venules and through medium-sized veins, and then through the large veins back to the heart. The capillaries are the only vessel that allows blood to exchange material with the interstitial environment. And this is because there's thin walls and there's pores, little holes, to allow for that exchange to occur. So you can pause the video here, go back and review. Let's talk about some disorders of our vessels. Arteriosclerosis. This is the thickening and toughening of arterial walls. The complications from this disorder result for about half the deaths in all the United States. The varied effects can be coronary artery disease, potential heart attacks, stroke. This is caused from the formation of fatty deposits on the tunica media of our arteries. So it's associated with the damage to the endothelial lining. Blood can't flow properly. This is the most common form of arteriosclerosis, atherosclerosis. It tends to develop in people with elevated cholesterol levels or lipid levels in their blood, so they develop this atherosclerotic, it's a hard word to say, plaque, and those fatty masses are basically restricting blood flow to a certain area. Elderly men, typically 50s and up, tend to develop these plaques. The risk factors include high blood pressure and smoking. Sad thing about this, half the deaths in all the United States, almost all preventable. This is all diet, exercise, lifestyle. Think about that. There's a plaque deposit in the wall of an artery. This is a coronary artery. Coronary arteries are the arteries that feed the heart muscle itself. That's a heart attack right there. How do we treat plaques? Well, we can remove and replace the damaged vessel. You can also do something called a balloon angioplasty, where this catheter goes in with this inflatable balloon and kind of presses the plaque against the wall. It's very effective in treating small soft plaques, and it's much easier than doing a whole vessel replacement because you don't have to worry about tissue compatibility, et cetera, and the procedure can be done outpatient. So you don't even have to stay in the hospital. You go in, have this little angioplasty done, you're out for the rest of the day. There's the catheter, and you can see the balloon going in and expanding, pushing the plaques against the wall, kind of smushing it back. You can pause the video here, go back and answer these 3 questions. Let's talk about how capillaries are put together, the capillary arrangement. They're arranged in what we call a capillary bed. The capillary bed has several connections between arterioles and venules. Think of like the root of a tree connecting to another root of a tree. And then in the middle, on those very distal ends of the roots, are the capillaries communicating with all of the soil that's surrounding them. It's pretty much what it is. The internal connection pathway is called a metarteriole. Has a little smooth muscle that's allowing the vessel to change in diameter, and it also allows for a variable flow in between our capillaries. So in other words, this part of the capillary bed can be innervated, and this part of the capillary bed isn't innervated or nutrified with the blood, so on and so forth. So that blood flow can be adjusted. There's little precapillary sphincters in there turning off and shunting the blood one way or the other. These rhythmic changes in vessel diameter account for something called vasomotion. Vasomotion is the pulsing movement of blood. Sorry, my phone's up. My phone was dying. I just wanted to plug it in. Causing this kind of pulsing movement of blood through the tissue. So it's pulsing over here and not over here. And then it's pulsing over here and not over here. And then it's pulsing a little bit in both directions. So that's done through the precapillary sphincters. Our capillary bed could receive blood from more than one artery. It could receive blood from what we call collateral arteries, where they fuse and give rise to other arterioles. It's called an anastomosis. An anastomosis is when one artery leads to another artery. Typically, arteries lead to capillaries, which lead to veins. Sometimes arteries lead to other arteries. it's called an anastomosis when that happens. That precapillary sphincter is going to control the blood flow into that capillary bed area. An arteriovenous anastomosis is the direct connection between an arterial and a venule without having a capillary bed. This would be used in times of sympathetic activation when you want to reroute the blood away from, say, the digestive tract and route it to the respiratory. So you're going to shut off those precapillary sphincters or shut the sphincters in the digestive tract and get that blood up to the brain or up to the lungs or wherever. So here you can see some of these terms I was referring to: collateral arteries, arteries leading into other arteries, leading into an arteriole. This is our overall capillary bed. The metarterioles are just little branches from that pathway, and precapillary sphincters are going to open and close different areas. So you can imagine this whole area might be shut down, but then this area would be open, or this area would be open, or vice versa. This is an arteriovenous anastomosis because we have one artery leading into another venule, and there's no capillary bed exchange taking place there. So if all of these precapillary sphincters were closed, again, maybe in times of sympathetic stimulation, if this was a capillary bed of the digestive tract, we would want to reroute all that blood back up to the lungs or the brain or the muscle tissue. You can pause the video here, go back and review. Let's talk about the venous system. The arterial system is typically under very high pressure. The peripheral venule pressure is much lower, about 10% of that of the aorta. So very low pressure. And so because it has such low pressure, we have to have ways of getting that blood back up to the heart. So there's two mechanisms that the veins use to get the blood back up to the heart. One is venous valves, and one is the skeletal muscle pump. So the valves are basically going to permit only unidirectional blood flow. So think of a long vein in your legs. That blood has to go back up your leg. So the blood's going to flow through a valve checkpoint and then stop. And then flow through a valve checkpoint and then stop. It's not going to go back down to your feet. That would be blood pooling. That would be dangerous, right? So if the valves don't work properly, that's when the veins can pull blood, and that's when you get varicose veins. This can also result in something like a hemorrhoid, like around your anal sphincter when you're pushing and there's lots of pressure, and that blood isn't permitted to flow up because there's lots of pressure and pushing going on there. Those veins around that area can get inflamed and be very, very uncomfortable. Here are those valves in your legs. As you can see, they're unidirectional valves, only allowing for the one-way flow of blood. Blood can be distributed unevenly between arteries, veins, and capillaries. In fact, the systemic veins are the majority of blood volume. About a third of the venous blood is in the liver, the bone marrow, and the skin. So liver, bone marrow, and skin have a huge blood supply. Think about it. Why would it need to be in the liver? Liver is our main metabolic center. Why would it be in our bone marrow? Bone marrow is where it's being made and recycled. And our skin, that is there for our first line of defense. So you want to keep blood to all three of those areas. Systemic arteries have a very small portion of the blood. Veins have the majority of the blood. The remaining capillaries, heart, and pulmonary circuit have the rest of the blood supply, a much smaller percentage. Here you see the overall distribution. Again, venous system, pulmonary circuit, heart, systemic arterial system, namely the liver, skin, and bone marrow. So let's talk about something called vasoconstriction. The vasoconstriction is going to maintain the blood volume in the arterial system, even in the case of the hemorrhage. So if you're bleeding uncontrollably, vasoconstriction is going to occur. It's controlled by the vasomotor center of the medulla oblongata. Remember, medulla oblongata is regulating respiration and heart rate. Sympathetic nerves stimulate those median muscles, resulting in reduced diameter of the veins and therefore restricted blood flow. You can pause the video here, go back and review. Let's talk about the circuits. I mentioned them briefly earlier. Let's talk about the pulmonary circuit. The pulmonary circuit is carrying deoxygenated blood from the right ventricle over to the lungs and then returning the oxygenated blood back from the lungs to the left atrium. The blood in the left ventricle is going to the systemic circuit. It's going to transport the oxygenated blood to all the tissues and organs, and then the deoxygenated blood is going to come back to the right atrium. So here's another overview of this pathway. Again, you can start anywhere. It's a circuit. So if you want to start in the brain, that's fine. Start in the brain. All the blood from the brain goes down into the right atrium. From the right atrium, we're going into the right ventricle. From the right ventricle, we're going to the lungs. From the lungs, we're going to the left atrium, then the left ventricle. Then we're going to go out to the aorta, and maybe now we'll go to the kidneys. And then we leave the kidneys, and we go back to the right atrium, and so on and so forth. So it's a pathway that just continually occurs. And at any stop along the way, there's blood in all of these places. So our blood vessels are all organized symmetrically. So in other words, you have right and left of almost everything. The largest vessels are near the heart. The smaller ones are in the periphery. There's only one vessel, but several names. Single vessel name changes happen when you change anatomical boundaries. So in other words, it's all the same highway. Think about a road where you live. Maybe that road changes names when you pass through a certain intersection. It's the same road. You didn't turn off the road, but now instead of being called whatever, it's now called something else just because you passed a certain boundary. That's all it is. It's the same thing in blood vessel world. You're on the same route. You haven't turned off anywhere, but because you're in a different region, you're now called something different. So an example here is the external iliac artery becomes the femoral artery. Then the femoral artery becomes the popliteal artery. And then the popliteal artery becomes the tibial artery, etc. A redundant supply and drainage to tissues can impact one single blood vessel. And anastomosis, which is a connection between adjacent arteries and veins, can reduce the impact of a blockage. So in other words, usually there are several routes to get into a specific area or out of a specific area. Let's talk about the pulmonary vessels. The pulmonary trunk is the large artery that comes out of the right ventricle, branches into the right and left pulmonary arteries, and the right and left pulmonary arteries branch into the pulmonary arterioles, which then go into the alveolar capillaries, where a gas exchange occurs. Then the oxygenated blood returns along small venules that join to form the pulmonary veins. The pulmonary veins, there's two right and two left, go into the left atrium. There you see it. You can see that exchange happening within the alveoli. You can pause the video here, go back and review. We're going to keep moving and shaking through the systemic circulatory system where we have systemic arterioles and systemic veins. All the vessels in the arterial system originate from the aorta. The aorta is the big vessel that branches off of the heart. Most of our arteries are paired left and right, and same thing in the systemic circuit or in the venous circuit. All the veins are going to drain into the superior inferior vena cava. Superior is the head, neck, and chest. Inferior is everything below the head, neck, and chest. Legs, abdomen, etc. Here you see all of the arteries, our arterial system. And major veins of the neck and limbs are located deep beneath the skin for protection because that area is not really protected by bones. So there's two sets of peripheral veins. There's one deep, there's one superficial, and those are important in controlling body temperature. In hot weather, the venous blood is going to dilate to allow extra heat to escape the body, and in cold weather, the venous blood is deep in the body, and those superficial veins are constricted to keep heat in. Here you see what I mean, and here you can see those deep veins and superficial veins. If you look right here, the brachial veins can be deep, the cephalic and basilic veins are superficial. You can pause the video here, go back and review. Let's talk about the branches of the aortic arch. And remember, I'm not going to give you a diagram on a test or quiz, but I may ask you the context of these vessels. So the aorta arises from the base of the left ventricle, and then it divides. It is divided into the ascending, the aortic arch, and then the descending. And you can see that very easily when you look at a heart model. Off of the aortic branch, there's three vessels. In order, there's the brachiocephalic. That's the first branch. That brachiocephalic is going to go right up, and it's going to branch into the right subclavian, and it's going to branch into the right common carotid. I just went on my left side. No, you can't really see my hands anyways. The right common carotid. Then you have the left carotid, just going to go straight up on the left side, and then the left subclavian, which just goes right down your shoulder. So the brachiocephalic trunk is going to subdivide and go to the right side, right carotid, right subclavian. Subclavian, below the clavicle, right? In the thoracic cavity, that subclavian artery is going to branch into the thoracic artery, which is going to the pericardium in the chest, and the vertebral artery, which then goes to the brain and spinal cord. After passing through the first rib, it then becomes the axillary artery because it's now out of the clavicular area and it's in the armpit area, which is the axillary. And then it goes and is now called the brachial artery as it's going down your arm. Then it's going to branch within the forearm to form the radial and ulnar artery, and then the palmar arches and digital artery. So again, just the same vessel, it's the same tube, it's just branching a little bit and entering different areas. And here you see that pathway. Here's our aorta, brachiocephalic trunk into our right subclavian and right common carotid, and then it's the axillary, and then it's the brachial, and then we have the radial and ulnar, palmar arches, digital arteries. So then we do our veins from the superior vena cava drainage. We have our external jugular vein going right into the superior vena cava, our vertebral vein, and our internal jugular vein, all going into the superior vena cava from the neck. Veins of the hand and forearm, from the digital veins, they're going to drain into the deep palmar arch, the ulnar and radial vein, same names as our radius. Then the superficial palmar arch are going to the cephalic, basilic, median antebrachial vein, a little bit more veins there. And then again, all just fusing ultimately with the brachiocephalic vein. There's right and left brachiocephalic veins going into our superior vena cava. Do you have to know all of these? I mean, yeah, I think you should. They're named after the regions of the body. They're not any trickier than knowing the bones or the muscles. Once you know the brachial region, you know what the brachial vein is or the brachial artery. So here you can pause the video, go back and review. Let's talk about the arterial supply to the head. There's the common carotid arteries, which branch into internal and external arteries, and they drain into what's called a carotid sinus. Then you have the vertebral arteries or the basilar artery, again, all up in the brain. And there they are here. You just have to know that those are the arteries of the brain. So I might say, which of these is not an artery of the brain? And I'll give you a list. And you just have to be able to identify which one isn't on the brain. Then all of the brain blood, so to speak, is all drained from the head. You have external jugular, internal jugular, and vertebral vein all coming back down into the brachiocephalic vein. And that brachiocephalic vein is going to drain right back into that superior vena cava. You can pause the video here, go back and review. Let's talk about some blood supply to the brain. I'm not going to get into all of these because, again, they're just named after where they are. Anterior cerebral, middle cerebral, ophthalmic. That's maybe one that isn't very commonly known. It's just the artery going to the eyes. Again, I'm not going to give you a diagram, so you don't have to worry about being able to identify them. Your lab instructor may. Then you have your cerebral arterial circle, which is sometimes called the circle of Willis. This is basically a protective mechanism, making sure that your brain is getting blood. If the brain can't come in through the carotids, it's going to come in through our interior intervertebral vein. So it's just making sure that they can come in through any pathway, and the circle of Willis is just kind of the backup plan there. Blood can drain through the vein through what we call sinuses. There's a superior sagittal sinus, which is a big kind of pooling area for the blood. It's all draining into the great cerebral vein, going into the cavernous sinus, the vertebral vein, all getting into the jugular veins, taking that blood back down into our superior vena cava. So you can pause the video here, go back and review. The descending aorta has branches off of it, and all the branches, again, they're named after where they are or where they go. Thoracic aorta branches into bronchial. They go to the bronchial, the lungs, right? Esophageal, mediastinal, pericardial, intercostal, phrenic. Phrenic goes to the diaphragm. And then the abdominal aorta with its branches. The abdominal aorta has the adrenal arteries, the renal arteries, the gonadal arteries, the lumbar arteries. Again, which of these is not off of the abdominal aorta? The adrenal, the renal, the gonadal, or the brachiocephalic, right? You should be able to tell by those names where they are relative to body position. Shouldn't trick you up. The superior vena cava is going to get blood from the azygos vein and the hemiazygos vein. That's all coming from the chest, the intercostal, esophageal, bronchial, mediastinal veins, all draining into the superior vena cava. Blood from areas below the diaphragm are going to go into the inferior vena cava. So those would be things like the lumbar veins, the gonadal veins, the hepatic, the renal, adrenal, and phrenic veins, all going into the inferior vena cava. Again, sample question. Which of these drains into the inferior vena cava? Right? Which of these drains into the superior vena cava? As long as you know thoracic and abdominal and those regions, you should be able to answer that. There's also blood supply to the abdominal viscera. These are our specific organs. So you have the celiac trunk. The celiac trunk's branching into hepatic artery, going to the liver, right? Gastric artery, splenic artery, the superior mesenteric and inferior mesenteric arteries, all leading into and supplying our intestines, stomach, pancreas, etc. Do you have to label them on a diagram? Not for me. You might have to for a lab. You have to ask your lab instructor for that. The hepatic portal vein, this is all, again, drainage. The hepatic portal vein is formed from the superior mesenteric vein, the inferior mesenteric vein, and the splenic vein, all coming together and basically coming back and fusing with that inferior vena cava. You can pause the video here, go back and review. Blood supply to the lower limbs. You have your right and left common iliac arteries, which is what your abdominal aorta basically splits into. And then your right and left common iliac arteries are going to branch into internal and external iliac arteries. The external iliac artery becomes the femoral artery, and the femoral artery, as I mentioned before, comes the popliteal, and then that is going to divide into the posterior and anterior tibial arteries, the fibular artery, dorsalis pedis, dorsal arch, plantar arch, and then it's all going to come back up in the venous system, where our plantar venous arch is going to get blood or give blood to the anterior tibial and posterior tibial and fibular veins. Again, just going right back up those regions, right back up those bones. Dorsal venous arch is going to drain into the great saphenous vein and small saphenous vein, into the popliteal and femoral veins, and ultimately coming back into the external iliac vein, where it joins with the internal iliac vein to form the common iliac vein. And that common iliac vein fuses to become the inferior vena cava. That's pretty much it. Pretty much it. That was a lot, right? But again, don't be concerned with the blood vessels. The most important part of this chapter is understanding blood pressure. It's understanding the flow of blood. It's understanding the difference between veins and arteries and how they work. And of course, the overall functions of blood. So let me know what questions you have. And I'm going to enjoy my little sandwich now. It's making me super hungry. And let me know what questions you have, and have an awesome day.